Okay, this is number 4, phagocytosis, slide 20. And there's a little video there embedded in the PowerPoint slides, so make sure you watch that. Just going to go through the stages of phagocytosis. This applies to things like neutrophils and macrophages. First thing is that the phagocyte has to get to the site. So chemotaxis, chemochemical taxis movement, chemotaxis movement in response to a chemical. We've looked at the action of C5a, which is an important chemoattractant. Once the phagocyte arrives at the site of infection, so you've got that splinter, you have the phagocytes arriving at the site of that splinter, getting out of the blood and into the tissues. They then need to be able to identify the bacteria in this case. They need to identify the foreign material, and they need to attach to it. There's a couple of ways that phagocytes can do this. First of all, we talked about the idea of a bacteria being covered in this sugar called mannose. And our phagocytes have receptors for that mannose molecule. So they can bind directly to the bacteria in this case, using their receptors, fitting right onto the mannose of the bacteria. The other thing we looked at is opsonization, where we've already covered the surface of our bacteria in some kind of substance. In particular, in the case of the innate immune response, we've covered the surface of a bacteria in C3b. And our phagocyte then has receptors for C3b that allows it to recognize the tagged bacteria and to stick to that bacteria and then move on to the next part, engulfment. Our phagocyte reaches out with its pseudopods, engulfs our bacteria and puts it into a little membrane bubble. That membrane bubble is called a phagosome. So here we have our bacteria. Here we have our, let's say, macrophage arriving, docking, maybe through the opsonization process of C3b. We have it extending its pseudopods, and eventually it'll extend its pseudopods all the way around our bacteria, and our bacteria will be placed into a little membrane bubble, and it's called a phagosome. So a kind of an eating vessel. Membrane, remember, is lipid bilayers. Whenever I use the term membrane in relation to a cell, I'm meaning a lipid bilayer. On to slide 21. Inside our cells, and this should be familiar from general biology, we have these little pouches of chemicals called lysosomes. And there's also little membrane sacs full of chemicals. We send in lysosomes. Lysosomes bind to the phagosome, and the addition of a lysosome to a phagosome produces this thing called a phagolysosome. So here's our phagosome. Here are our lysosomes, full of their digestive chemicals. These are going to arrive. They're going to attach, and all of the chemicals that they brought with them will be put into the interior of the structure And now that our lysosomes have stuck, this whole thing is now called a phagolysosome. Lots of things occur in the phagosome and in the phagolysosome. What happens to the particle that's been ingested will rely in a lot of ways on the toll-like receptors. So toll-like receptors on the outside of cells like macrophages, but also pointing into the inside of a phagosome, and then also in our phagolysosome, because the inside of our phagosome was once the outside of the cell. Often the pH lowers, so it becomes more acidic inside the phagolysosome. So once you have your bacteria inside your phagolysosome, you have then destruction. You're trying to kill that bacteria. The lysosome itself brings with it many digestive enzymes. So digestive enzymes are enzymes that take large molecules and snip them up into smaller pieces. For example, they might start to digest the peptidoglycan, destroying the cell wall of our bacteria and causing it to burst. Other destructive chemicals in our phagolysosome include these things called reactive oxygen species. So we have we have our reactive oxygen species. Oxygen is a very reactive molecule, and the products of working with oxygen are typically very toxic. And all of those toxic chemicals are dumped into the phagolysosome, and they then destroy many elements of the bacteria. Things like it'll destroy the function of proteins and destroy the function of DNA. We also dump in there things like antimicrobial peptides, and we dump in there our lactoferrin, which we encountered before. Lactoferrin sucks up all the iron that may be still available to the bacteria. The bacteria's biochemistry relies, as do all cells, relies upon iron. So if we deny that iron to the bacteria, it can't do its biochemistry. Once we have then destroyed the bacteria, we are left with our phagolysosome full of the bacterial pieces, and we then expel those pieces out of the cell through a process of exocytosis. So we have all of our chemicals coming to bear here. We have the destruction of the bacteria. We have all of the cell pieces, and then our phagolysosome will bind to the outer surface of our cell, not quite to this extent, but you get the idea. And then those destructive pieces, those waste pieces, are expelled into the environment through a process called exocytosis. Exo means outside, so cytosis means the process of expelling material to the outside using a vessel. Slide 23 has a picture of all those different components. Number one, chemotaxis. We have to get the phagocytes to the site. Number two, recognition and attachment. They have to identify the bacteria and stick to it. Maybe they'll stick directly to the bacteria by identifying mannose, or they'll stick to the bacteria because it's been tagged with, say, C3b. They'll engulf that bacteria into a phagosome. they'll send in their lysosomes full of chemicals. The phagosome and the lysosome will fuse into a phagolysosome. All of those nasty chemicals will destroy the bacteria, and then the pieces will be ejected through a process called exocytosis. So I've used a bacteria as an example throughout these processes, but a phagocyte might eat up all kinds of different things. It might eat up a virus and go through the same process as destroying that virus and spitting out the pieces. Okay, and that brings us to our study guide, and that's where I'll stop this video.